Hi, everyone, and welcome. I'm Scotty Sims, one of the founders and executive director of KCNQ2 Cure Alliance. I'm also a KCNQ2 mom and really glad you're all here. Tonight is all about how families can directly help move research forward through our biospecimen donation. We're joined by Dr. Lindsay, I'm going to mess it up,
Zoric.
Zoic.
Yep.
I got it. From Combined Brain. Combined Brain connects more than 130 rare neurological disorder communities with researchers, clinicians, and industry partners to help turn data and samples into real research projects. Lindsay has over 20 years of experience in biomedical research and is also a rare disease parent herself. She brings both experience, deep expertise, and lived experience to work. So before we get started, just a couple housekeeping notes. Keep all your microphones muted. If you haven't already, feel free to open the chat. You can drop questions in the chat at any time, and we'll come back to those at the end of Lindsay's presentation during our Q&A session. If you'd like to ask a question out loud, you can always use the raise the hand feature. It's the button at the bottom. I think it looks like a heart. And raise your hand and we can call on you. We are recording today and we'll be sharing the recording afterwards on our YouTube channel so you can watch it again or pass it along. Lindsay will present about 20 minutes or so and then we'll do our Q&A. All right, Lindsay, it's all you.
Wonderful. Well, thank you guys for having me today. Let me get my slides up here. Can you see the whole screen? Okay, awesome. Well, thank you guys, and thank you for the lovely introduction. My name is Lindsay. I'm the CSO and the Biorepository Director at Combined Brain. I've been with the organization for about 6 months now. It's been a group that I've worked with for a while, prior to that too, with the SYNGAP Research Fund. So just to give you a little background of Combined Brain, like Scotty mentioned, we're here to create fast-track therapeutic development for rare neurological disorders, and our leadership team are all rare NDD parents. So Terry Jo has a son with Angelman's. I have a daughter with SYNGAP-related disorder, and our Chief Operating Officer Taylor has a son with CHD6 related disorder. So just to give you a quick overview of the translational research pipeline, and I'm going to be a little bit quiet for a second because my daughter's coming in, and if you have kids with rare disease, you know they can get a little crazy. So we work across multiple different uh, uh, research approaches within the pipeline, from diagnosis, discovery, development, clinical trials to approval. So advancing new therapies requires a large translational pipeline. A lot of times this is a difficult lift for individual patient advocacy groups. So the goal of Combined Brain is to kind of create a shared infrastructure that all patient advocacy groups can use together. So while pharmaceutical companies are increasingly engaged in rare disease drug development, the disorders often lack early commercial investments just because it is kind of rare. There's not a lot of patients. Those patients are spread out globally, so it's difficult sometimes to study them in a localized way. So PAGs can lead that early stage development by generating data, developing infrastructure and momentum that helps industry engage and kind of de-risks therapeutic development. So we support that infrastructure across those therapeutic pathways. And these are some of the different studies that we have that we work with that do that specifically. Those that require more of the patient samples and data to move those stages forward are highlighted here. And this is where the KCNQ2 Cure Alliance Biorepository comes in really helping move forward discovery, develop, and advance clinical trials through the creation of disease models, biomarker discovery, multi-omic studies with phenotypic research, developing advancing different research projects like drug repurposing, observational studies with scientific collaborations and pre-competitive research, and help with clinical trials by establishing natural history studies, outcomes research, advancing ICD-10 codes, and just getting the groups organized and ready for clinical trial to reach that approval stage for final drug development. So today I'm just going to touch a little bit more on two phases of the work that we do, that the KCNQ2 group has been involved in biomarker discovery and pre-competitive research. Before getting into that research results specifically, I just want to talk a little bit about the biorepository and what it is. So within that whole structure of the pipeline of translational research, you need to be able to have a centralized resource for rare neurological disorders to have these samples available for your community to be able to access and do novel research on. So what the Combined Brain Biorepository is, it's that centralized research resource for rare neurological research, where we coordinate the collection, storage, and distribution of biospecimens. Our collections are all occur under an IRB approved protocol that ensures ethical oversight and protection of our participants. Your patient advocacy group retains the ownership of those samples. So what we provide at Combined Brain is the centralized infrastructure. The samples are all housed in at Van Andel Institute. It's a nonprofit organization out of Michigan. We provide operational standards, storage partnership, and research access management, meaning if there's a request for those samples, we will execute all the agreements so that those samples can be used ethically and effectively. We vet qualified researchers that can access specimens through a structured process. All of their requests are approved by the patient advocacy group prior to distribution. So the governance of those samples is maintained by your patient advocacy groups, by your group specifically. And if you have biospecimens stored in other locations, those can also be transferred into Combined Brain Biobank. So if you have worked with researchers in the past that have developed specific cell lines, if your researchers have tried to share those with other academic institutions, maybe that can be a very difficult process. But you as a patient advocacy group can centralize all of those resources in this KCNQ2 biobank, and you can then distribute those more easily across different research partners. So how the biorepository works, we work to collect samples. So samples are collected through home phlebotomy, event-based roadshows, clinical visits, or institutional transfers. We process and store those samples. So depending on the types of samples that you're interested in incorporating into your biobank, we do most of our collections are flash frozen, which means that they're just immediately frozen after collection, which is kind of the best standard for biomarker development. And then they're shipped to a secure research facility that we contract with. We have the request and the review process. So researchers submit formal requests for samples, which we review those requests for consent alignment, and then your community approves those requests or denies them based off of your research goals. And then we distribute those samples by executing distribution agreements with the qualified academic or industry investigators. So just to basically touch on the samples that are available in your biobank already, you all have a really lovely biobank available of controls as well as KCNQ2 related disorder in your biobank. So this is the number of participants. You have blood spot cards, plasma, urine, and whole blood that's available across these different individuals. I'm just going to highlight that you have 23 plasma samples from 23 participants, and all of the samples are available from a single time point. How these biospecimens can be used for research. So there are community biospecimens that can be used for various different studies. So really any way that you want to leverage these samples to better understand your disorder. That could be through different academic or industry research partnerships that you have. You can connect with them, you can send them samples, they can request samples from the biobank just to be able to study your disorder in greater detail. Combined Brain also has service agreements with different operational partners to facilitate access to different research. Like I mentioned, we have master service agreements with different functional partners, whether that's to create iPSCs, different stem cell lines or neurons, a lot of different ways that you can approach research through Combined Brain. And then we also coordinate pre-competitive and grant-based research, which is at no cost to the patient advocacy group. It's from funding that Combined Brain coordinates either pre-competitively with industry or grant-based based off of different research grants that we have either from different sources like NIH or CZI, the Chan Zuckerberg Initiative. So just to give you guys an idea of how specific types of biospecimens are used, because I know sometimes it's difficult to know what to collect based off of what type of research that you're interested in, or what type of work that researchers might be interested in doing on your disorder. So here's some of the biospecimen types that are commonly collected: PBMC, plasma, whole blood urine, or nasal swabs. These are the most common types of samples in our biorepository. If you have PBMCs in the biorepository, you can do development of stem cell lines. That's what this the iPSC stands for. And those can be differentiated into neurons for different research. There's different access to different cell culture or functional studies if you're interested in kind of looking at the effects of specific drugs on your target cells. Biomarker discovery, being able to understand what proteins are expressed differently within your disorders so that you can track that over time as a measure of potential future treatment efficacy. And that can be done with a lot of different plasma samples. And then another group that we work with, which is very specific to the nasal swabs, is the rare shift drug discovery with Unravel Biosciences. It's specifically for doing drug repurposing. So sometimes there are disorders that really don't have effective treatments. And in that case, we work with a group that will do transcriptomic based omics studies to determine which drugs might be effective at helping the disorder in a therapeutic way. So to just kind of highlight some of the work that's been done with your biosamples is a biomarker discovery using the 23 plasma samples that you have available in the biobank. And this was a pre-competitive project that we executed a few years ago now, and we're just kind of finishing that data analysis and really reviewing everything that we've done so far so we can come up with our next series of studies. So I just wanted to highlight that because sometimes I feel like samples go into the biobank, you provide samples, it can be really difficult to even, get your kids to comply with providing samples, even getting your kids, I know for my daughter, getting her to a collection site is really difficult. So it is a challenge. And then it's sometimes not easy to see what happens after that. So just to give you a bit of an idea of what happens with those samples, so we established a Combined Brain, we established a pre-competitive neurodevelopmental disorder plasma proteomics study. And this was the first study that we did in this way. And the goal of this study was to determine if there were any disease-specific or cross-disorder differentially expressed proteins. And a differentially expressed protein is really just something, a marker that's different in the affected person as compared to a healthy control. Or looking for dysregulated pathways. So something that might be off as far as an immune system might be a little bit different this way or that way, or different types of inflammatory pathways or something like that. If you think about certain pathways that might be triggered as opposed to specific individual markers. We wanted to know if those could be identified through a cross-sectional plasma proteomic analysis. So a cross-sectional plasma proteomic analysis is just taking those 23 plasma samples that you had from your individuals and looking at to see what proteins are in those plasma samples. And we did a cross-sectional profiling of 11,000 different proteins. And this was a study where we evaluated plasma samples from 520 individuals with rare neurological disorders and 173 to controls. So it was a very large study that included 29 different disorders, including KCNQ2. So that was a large assay where we looked at those 11,000 different proteins. And how our pre-competitive projects work is that we work with industry partners that want to be able to develop biomarkers for the disorders that are within Combined Brain. So those industry partners fund the research, which is a great opportunity to be able to kind of explore these things without having to pay for the assays themselves, which can be expensive. So we have industry partners that pay for that work and continue to drive the research forward. We work with patient advocacy groups, so with your organization, to prioritize some of those research questions. And then as your patient advocacy group has different academic partners, we can also work and share data throughout this partnership. So this kind of becomes a data working group. And what we've just done, completed over the last 3 or 4 weeks, is a summation of this entire study. We had all of our partners in the room, academia, industry partners, and our patient advocacy group leaders, to break this data down, determine what were the most important results, and how we can move forward with that in the most meaningful way. So when we're looking at these protein profiles, we want to be able to create biomarkers for clinical trials, understand disease biology, and we can use that to identify novel treatments. So results from that study look a little bit like this. This looks a little less clear now that it's on the big screen here. But so the goal was to identify, like I said, proteins that were expressed differently within certain disorders as compared to healthy controls. So there were certain proteins or markers that were upregulated, so disease-specific upregulated plasma proteins, or proteins that were downregulated. So this is the profile specifically for KCNQ2 related disorder. These are this proteins, which they always kind of just look like a cluster of letters, but there was a cluster of proteins which were downregulated and a cluster of proteins that were upregulated. So if we're able to compare these across a larger number of participants within your group, that would be a signal to be able to pursue for biomarker development. And kind of what that looks like in thinking about this cross disorder, this is just some of that same data. So one of the downregulated proteins within that list was a protein called activating transcription factor 6. One of the upregulated proteins was an enzyme called a methyltransferase. So this is just a very small snapshot of that very large data set, just to give you two specific examples of how your disorder may look like other disorders. So these are all disorders that have that are characterized by epilepsy. So within this disorder, you had your disorder, KCNQ2, had a reduction in the overall expression of this protein, the activating transcription factor. So there was a reduction in this protein as compared to controls, and an increase of this protein as compared to controls. So the activating transcription factor is a protein that helps clear damage in unfolded protein. So having a decrease in that just indicates that there's a decreased ability to handle stress within the cells. And this is really something that we're seeing across multiple different neurological disorders. Most all of them that are shown here have that same profile. So the cells are within these disorders are kind of acting in the same way. That may mean that there's a certain treatment pathway that could be targeted for these different disorders. And similarly, there's an increase in some of these different transcription levels. So this is just a snapshot. This is not meant to be a comprehensive analysis of the data, but just to show you kind of how we're handling it, how we're looking at it. And there is a publication coming out from Combined Brain that we plan to submit in the next month or so, which is going to recap all of this information, and provide a platform for our future studies. So from all of the analysis that we've done, we can say that there is very high variability in proteomics or in biomarker profiles with individuals within a specific disorder. So what we hope to do for future studies, and what we've discussed with all of our stakeholders, which industry, academic, and PAG leaders, is to prioritize longitudinal assessment to resolve those meaningful biological signals. So longitudinal meaning if there is a participant that's donated this year, have samples collected and analyzed from a year later. So kind of continuing to follow that participant as they age to determine whether the signals that we find within the single time point, whether those are maintained and are stable throughout the development process. Age was one of the main factors that we determined as being responsible for impacting all protein expression. Age, sex, as well as different clinical subtypes. I'm not as familiar with KCNQ2 for instance, SYNGAP has a lot of different mutation types, and that can present differently, different levels of severity. So one thing that we did to try to resolve that analysis was to stratify those subgroups, looking just at the pediatric population or just at adult population, just at males or just at females. And that did improve some of the resolution and the kind of the ways that we can understand these proteins. But when we do that, when we start to look at those subgroups, that means there are less participants overall to be able to analyze. So when we think about improving that signal, that means we need to include more participants. So for those two statements, if you haven't donated samples already, you can donate again to support the work and the evaluation of longitudinal samples of multiple time points per participant. And if you haven't donated samples yet, you can donate those samples, and those results would be added into this analysis to try to improve the resolution of the results that we already have. So we're also continuing to follow up on the different pathway level and multi-protein biomarker strategies, just because those do offer some insights into different treatment approaches, which could be valuable to your community. So just to mention too, if there are researchers within your community that would like to access this data to be able to continue to analyze it in ways that's meaningful for you, we just need to sign a data use agreement, and your research community can have access to this data set. Okay, so that being said, those are the types of research that are going on at Combined Brain. Those are the some of the research that's been done at Combined Brain with KCNQ2 specifically. So how can families continue to contribute to this? So families really play the most vital role in advancing research. Many of the insights that are needed to advance treatments for these rare disorders are understood through evaluating the affected individuals. So by contributing those biospecimens to your biorepository, you can help better understand the neurological disorder and help accelerate those treatments. So there's multiple ways that you can donate samples if you're interested in doing that. You can participate in a roadshow. Roadshows are kind of collection events that are held at participant community gatherings, whether it's a conference or just a kind of a family meeting or a science meeting, any different type of event where you have families getting together. We can meet your community there or meet a community there and collect samples from participants. And this is just a great way to be able to do it in a convenient and supportive environment. Sometimes it's a little bit easier for kids if they see other family members or friends that are undergoing the same types of collections. But we can also schedule home collections if you are interested in contributing but just really can't attend those events. Totally understandable. And for those instances, we can coordinate mobile phlebotomy services where we have a trained professional visit the home and they collect samples that your community is interested in exploring. And then we just ship those samples back to the biorepository. And if there are certain collections, certain procedures that your patients are undergoing, like tethered spinal cord release is an opportunity to collect cerebral spinal fluid. A lot of our biomarker studies have been done with plasma, but the cerebral spinal fluid is probably a little bit more reflective of the neurological state within these disorders. So that is a lot of researchers are interested in evaluating CSF. So there are certain procedures where CSF is collected and just normally discarded. And we can work with your physician if you're having some of those surgeries to be able to collect that sample and then send it back to the biorepository for future research. So any of these sample donations can be scheduled with Combined Brain through your patient advocacy group. So a little bit about what we collect. We'll do a lot of things. Combined Brain is really here to serve patient advocacy groups in whichever way they need. Most of the things that we mainly collect are blood, and that blood can be processed into plasma or PBMCs, which are just peripheral blood mononuclear cells. They're just the cells that are circulating within the blood. We do different swabs for different things. It's just an easy way to access DNA and RNA. So buccal swabs, cheek swabs, or nasal swabs. Saliva is also just an easy way to access DNA. Urine for different metabolic studies, stool for different microbiome studies, and then also those physician-based collections, whether that's a tissue biopsy or cerebrospinal fluid or something like that. Okay, so just to give you an idea of what happens when we're collecting these samples, so what happens before collections, this is probably the biggest lift in getting these things done. So if you're interested in donating to the biorepository, so before your collection, you just need to let your patient advocacy group know that you'd like to donate. They'll contact us and give you your information so that we can contact you to get this set up. So for either if you want to participate yourself or if you have an affected family member, we can do affected individuals as well as unaffected controls. Then a combined brain research coordinator will reach out to you to help you create a clinical research ID or a CRID, which is basically just a de-identified code that helps maintain your privacy. So there's no names or anything that's associated with your clinical samples. And then a matrix account too, which is a platform that we use that has all of the consenting forms and any other surveys that your community might be interested in evaluating. So there's different things on the matrix portal that your patient advocacy group might have you fill out, whether it's reports like a seizure survey, or everybody's probably familiar with the Vineland. I know that's a very difficult one to even think about taking. But those types of things are very helpful in being able to associate with the clinical severity or genetic variants, different clinical factors like that can be associated with the samples. So when we're doing a different, or when our researcher is doing any type of analysis, that information can be used within the analysis. So we use that platform to collect those surveys as well as assign the consent form for the donation. And then all those surveys are completed, anything that's requested by your patient advocacy group. So if you're collecting samples at a roadshow, so you just need to, once you're at the roadshow, you have your consent created, everything's done. You just need to check in with the Combined Brain staff at the roadshow. We have a check-in table right out front. Our staff will review your paperwork and ask you a few questions. So there's a few questions that we do day of, just to make sure that there's anything that hasn't been updated, whether that's any recent seizures, any medication changes, those types of things. We want to be able to capture that, anything that would directly affect your collection for that day. And then we have trained phlebotomy members on staff that will help collect samples. And that collection typically takes like 15 to 20 minutes, depending on what type of samples are collected. And that's it. Then we process the samples on site and send them back to the biorepository. If you're doing a home collection, it's kind of a similar procedure, but we're then going to be working with the Combined Brain staff to identify a tentative collection date. And we have trained phlebotomists that we contract through an organization called Travel Lab. So they will be the ones that actually confirm that final date with you and then send the phlebotomist out to your home to collect and process the samples. So a phlebotomist just schedules an appointment with you, comes to your home, they bring all the equipment that they need to process the samples directly on site. So that process takes a little bit longer, 20 to 30 minutes, just because there's a little bit more setup. And again, depending on what samples you're collecting, the phlebotomist in that case will review all the paperwork and ask you some additional questions. So this is an opportunity that we have with our roadshows. So your community doesn't have to specifically have your own event. Your community can attend any event that Combined Brain is hosting. So if you don't host an event every year, you can attend any of these event locations by just telling your PAG that you're interested in participating at a specific event. They fill out a form online to let us know that you're interested. We will then go through the entire process that I just mentioned of reaching out to you, making sure that you have your clinical research ID and consent for the collection. Then you just attend these different locations or come to the different locations. All of the locations are available on our website. This is just an overview of the different cities that we're going to be in and when we're going to be there. So this weekend, we're going to be in Maryland. If anybody wants to stop by our Maryland site, we'll be in Elcott City and collecting samples Friday and Saturday. And these are the rest of the dates and locations that we have. There's a few more that might be added to the list. But this is an opportunity to just do a free blood collection. If we do if your community hosts a specific event, there is a cost associated with that. If you do home collections, there is a cost associated with that. If you drop into any of these road shows and donate blood, there's no cost associated with that. These are just free for the community to be able to, collect samples in a cost-effective way. So check out the schedule there and let your patient advocacy group know if you're interested in donating at any of those locations. That's all I had. I will stop there and would love to take any questions if you guys have them.
Yeah, Lindsay, thanks so much. I think one thing that would be helpful is for families to kind of understand, for those who might not know, is kind of what are biomarkers?
Yeah,
Why is the research so focused on that?
yeah. Um, So biomarkers are just it's something that stands out from the profile from your say affected child compared to your maybe typically developing child. So a biomarker is just a way to be able to identify something that's different that kind of stands as a marker of that disease, and it's called a biomarker because it's just a biological marker that you can identify for that disorder. And a biomarker could be something that you is not necessarily unique to your specific disorder. It could be a marker of, say, epilepsy. There are certain biological pathways that are triggered when there is an epileptic event. So you can find biomarkers for these different things. So a feature of the plasma that's just a little bit different because something is off with, or something is different with the biology, whether it's a seizure or whether it's a specific disorder like a KCNQ2. There can be disease-specific markers like a SYNGAP marker or a KCNQ2 marker. They both might have the same epilepsy marker, but differentiating between those two disorders, they would also then have their own unique biomarkers. So biomarkers are really helpful for diagnostics, specifically if you're interested in, not interested in, but I know many of you probably had a challenging diagnostic journey, depending on what you've gone through and how your disorder progresses. I mean, we went through diagnostic purposes for years just because there is no way to identify, besides whole genome sequencing at the time, to identify a specific disorder. So if there are clinical biomarkers, something that really differentiates your disorder from other disorders, that makes the diagnostic journey much shorter and much less painful. Those biomarkers can then be used to monitor effective therapy. So if there's a biomarker or a protein that's higher, like what we showed here, these biomarkers are upregulated. These proteins are upregulated in your disorder, or versus ones that are downregulated. So when you are receiving treatment, the goal of the treatment is to convert those back to looking like a normal, healthy individual. So having no proteins that are, or fewer proteins that are out here in this significantly dysregulated area. So being able to measure the more becoming more normal profile for those biomarkers is one way that we use to evaluate clinical effectiveness of different drugs, which is important for getting treatments passed by regulatory approvals.
Brooke.
Yeah, no, I was just going to say, Lindsay, thank you so much for that answer and for this presentation. What a really great summary and overview. And so, for those who don't know me on the webinar, and sorry for those who keep coming to the webinar and have to keep hearing my introduction, I'm Brooke Babineau. I'm the scientific director for KCNQ2 Cure Alliance. Been with the group for almost a year and a half now. Pretty soon I'll start saying a year and a half. And I've been really excited that we've been doing these webinars more frequently, and really excited to have Combined Brain come and talk about their program because it's something we've participated in for a long time. I'm glad to be able to kind of highlight the work they've done. And Lindsay did a great job showing you, like she just said, like these are people's data, and we are putting it to use to help better understand these rare disorders where very limited information exists. I'll try and do a couple quick points. One, I do want to highlight, there's a question in the chat about how long does the roadshow process take? Like if someone needs to kind of manage their day and they're going to take a 15-minute drive over to one of the sites, how long might they have to budget for being there?
If you have your consent done, if you have very limited time, sometimes the road shows can be a little chaotic, and depending on the sensory needs that your family has, try to do the consenting process before time, just so you don't have to sit there and do that on the phone. The first road show that I went to before I was with Combined Brain, I didn't do it, and it was just a bit of a pain. So the consenting process itself takes about 20 minutes. The blood draw itself will probably only take 15 or so. So if you get there, plan for 20 minutes on site just to do the blood collection. And, depending on what samples that you're giving, it goes really quickly. Depends on how difficult it is to draw from the participant.
Great.
And
Thanks.
I'm sorry, I was going to say, I dropped a link in the chat. It goes to our website and it breaks down the process of how to apply for the roadshow. So you can just email us at research@kcnq2cure.org. It has the information that you need to send to us. And then we fill out a form that generates, goes to Combined Brain, and then they reach out to you. So you can't just show up at a site. You have to have the consenting done ahead of time. So if you're looking to do one around, it's in the next couple days or weeks, whatever, please get that email to us. And that way I can go ahead and get that form submitted to Combined Brain. But the way the road shows work is some of them do fill up ahead of time. You'll see that they only have one on the West Coast this year in California, and it's already full. So if there's a roadshow that you think you would want to attend, I would recommend going ahead and filling out sending the email to us with the required information and getting your consent forms and everything done ahead of time. That way you're in their system and you don't miss out if they fill up ahead of time. Were there any other questions, or Brooke, did you have anything else?
I had two other quick ones if I can. And then so Lindsay, you pointed out we've had 23 different participants so far, and hopefully maybe we'll get some more in this coming year. But I also noticed, because I have access to our catalog of samples, that those participants, they have multiple samples, right? And so we can actually you showed one piece of data from a study we're participating in, but we also get requests from industry and things like you've mentioned, and samples will go to other projects as well. So I was just kind of wondering, I don't know if you can quantify, like, one person can provide multiple samples. I mean, what, like, each time you donate deposit samples, I mean, how much research could that potentially contribute to? I mean, is it like dozens of studies, or what does that kind of look like?
Yeah, that's a great question. And we try to maximize the number of available studies by making multiple aliquots. So for instance, if you come to a roadshow and you donate blood and we're processing that into plasma, we'll try to freeze as many individual aliquots as we can. Usually that's about 10, depending on how big the kit is and how much blood we get. So for one specific draw, we can usually get like 5 to 10 different aliquots of plasma. So that can be used for a lot of different research studies. So the research that we did for this pre-competitive project, they used the same aliquot for 3 different types of studies. Not all of them were shown here just for the sake of time, but so that's a great point, that one single donation can lead to a lot of different research opportunities, whether that's even different sample types too, whether that's whole blood, which can be used for whole genome sequencing, or PBMCs for transcriptomics. So there's just a lot of research opportunities that can happen with one single collection point. Yep.
Excellent. And then my last, hopefully quick question is just the physician coordination. I hadn't actually appreciated that point before, but what a good sort of efficient use of procedures that are probably not fun for anyone, but if you can
Right.
get some data out of it. And so was there a particular process with that? Would someone just again kind of reach out to Scotty, prior to that appointment coming up, and Scotty connects
Yep.
them with you? Is that sort of the process there as well?
Yep. So
Okay.
those are a little bit more delicate. It's not I mean, there is a standard procedure, but every physician is a little bit different, and what they
Okay.
can do is always a little bit different. So if you have a procedure coming up, just, let Scotty or whoever know from your community. They'll connect with us, and we connect with your research team and or your clinical team, to be able to coordinate that. And that can be anything from, we just helped a team with an aortic valve replacement, and now their heart tissues banked in the bank if they want to do different research with that. Um, and I mean, a lot of those things are just thrown away otherwise, and they can be valuable research for research. So yeah, we're happy to coordinate and make those connections. And I'll also
Fantastic.
say, and no, it's not fun to think about money when it comes to sample donations, but the biorepository is set up in a way that there's a distribution fee for researchers and industry partners that are interested in working with these samples. So if an industry partner is asking to get a sample, or some of that CS stuff that you just collected, and it's in the biorepository, 75%, the majority of the money goes back to your patient advocacy group. So it's just a way to make it sustainable for you. So if there's samples that requested, that means that is funding your organization.
And I'd like to
Excellent.
add on to that too, is, the more robust our catalog is with samples, the more appealing that is to industry, because they see that they have a great cross section from us. So if our sample sizes are small and limited, we're less appealing. Um, and we did have another question in the chat that asked about which option works for the drug repurposing, and are you notified when a researcher extracts your data?
For the drug repurposing, we um, which works for drug repurposing. So for the drug repurposing, there's a lot of different ways that you could do drug repurposing. The one study that we used, or that we have a master service agreement with Unravel. So we create master service agreements with these different organizations that do kind of basic research. And then the patient advocacy group just signs on a smaller agreement directly with Combined Brain to make it easier to access that. And we usually provide discounts for those two. Those are with nasal swabs, if that was the option for that. So we use nasal swabs, and that requires a same-sex control for the participants. So that's kind of a family matched control when we compare what a normal an unaffected transcriptome, which is just the RNA that's produced from the DNA, what a normal transcriptome looks like versus what kind of the KCNQ2 transcriptome looks like, and try to figure out ways to try to make that more normal, trying to realign that individual through drug treatment with a more healthy looking transcriptome. So those are nasal swabs. And are you notified when researchers extract your data? No, your patient advocacy group is notified when your samples are requested, and they have to approve the use of those samples. So that's the approval. We are trying to work with our research groups so that the results are going back to the community. So whenever that's possible, we try to make those results available to your community. That's not always feasible. Sometimes there's industry partners that request your samples, and we cannot disclose that, just because we're under NDA with different research partners, industry partners, that don't want to disclose their research interests.
Were there any other questions that we have? Okay. Lindsay, I want to thank you and everyone who joined today. I think this was really helpful, given a clear picture on something that families can yet another option to do directly to support research. This webinar has been recorded, so we'll have it up on the website. So if you want to come back to it, you can also find the blog on our website that has all the link that I put in the chat that has the details about the roadshow. And if you can't find that, you can always shoot me an email and I can get you connected with the whole process. But, you know, we've been working with Combined Brain for many years now, and the roadshow has been a really great addition to a service that they offer. And there's just no other way that a foundation our size would be able to coordinate something this extensive. So we really do ask our families to take advantage of it if they can. And please reach out to us at the foundation for any support or guidance we can give you to help kind of do the participation piece. And just know that participating year after year after year is really helpful because it does give that longitudinal data. So while we don't host a conference every year, we're biannually. On the off years, I sign our daughter Harper up and we go to a road show because we're in Denver and they usually have one here. We'll go to a road show in Denver, and that way she's keeping her data in the system, and it's helping build out this nice little picture of all her stuff. So thank you everyone so much for joining. Have a good evening, and let us know if you have any thoughts on topics you'd like to see us host in the future. Bye.
Thanks for having me, and thank you for all you do.
Yeah, thanks.
Bye, guys.
All right, bye.
Thanks, Lindsay. Thanks, everyone.